So this is your final topic. This is headache pathophysiology. And we are really only going to talk about headache because it is quite a large subject. So there's not an awful lot else we can fit into this session. But we will talk about all of the main primary headaches and one secondary headache. One of those primary headaches being migraine, which is a huge topic area with lots of theories about the pathogenesis. So we'll cover that in this part, and then in part 2 we'll look at some other primary headache conditions, but also look at the applied pathophysiology. Okay, so in terms of classification, we can classify headache syndromes into primary and secondary. Primary is basically where they are syndromes in their own right. There is no other cause other than the pathogenesis of that syndrome itself, whether we know it or not. So our primary headache syndromes are migraine, cluster headache, tension type headaches. There are others, and those you can look up in the International Headache Classification. I've given you the link there, and I'm sure Kerry will give you the link as well. Secondary headaches are headaches that are caused by other things. So we're going to talk about in part 2, well, a little bit what is known about medication overuse. But again, it raises a lot of questions. But other symptoms, other syndromes that can cause a headache, of course, sinus infections, sinus headache, very nasty. Or any systemic infection, because we have the release of cytokines within the brain, so they can cause headache. Obviously, infections within the central nervous system itself, so meningitis, encephalitis, those sorts of things. Trauma, so head injuries or bleeds into the cranium. Remember, it's a hard box of a system, so anything bleeding into that is going to exert pressure. Tumours, again, are going to exert pressure, or aneurysms. So these are sort of headache as a symptom of something else, not a syndrome in its own right. But the big issue here is, of course, and you may have seen images of people having brain surgery and they're still playing a guitar, playing a violin, playing chess. They are totally, the central nervous system is insensitive to pain. There are no nociceptors within the central nervous system. Of course, it's generally all about transmitting pain. It's the transmission pathways. It doesn't do transduction, the bit, the nociceptors. But of course, a headache is pain. So where's it coming from? So headache, or cephalgia to give it its proper name, involves activation of the trigeminal nerve. So we're talking cranial nerve 5. And the trigeminal, of course, has 3 big branches, and you can see the spread there in the diagram across the head. It's more about the little branches that go into the head that we're interested in terms of headache. But what stimulates the trigeminal pathway does vary in different headache syndromes. So the pathogenesis of a particular type of headache will have a different way of stimulating the trigeminal nerve to give headache. To understand the pathogenesis, though, so before we can even start to look at pathogenesis, you have to understand the trigeminal nerve pathway. The ascending pain pathway comes with that, but also its interaction with the meningeal vasculature. So the trigeminal nerve, cranial nerve 5. has three branches that we said. They repeatedly branch, and of course after they branch they get a different name, but they all originate from the trigeminal nerve. They become the sensory nerve, so they're primarily afferent for most of the head and face. The only efferent component is that they supply motor innervation to the muscles of mastication. Careful how I say that. Chewing. So the three branches, the V stands for 5 in this case. So we have 5-1, if you like, is the ophthalmic nerve. So remember, this is afferent. It's sensory from the eye, the nasal cavity, and the skin of the upper face. In other words, the green area that you can see in the diagram there. Our second branch is the maxillary nerve. That's our pink block of colour on that diagram. Again, sensory. Importantly, sensory from the meninges and the lateral midface. Our third branch is the mandibular nerve. So this is sensory from the dura, but also from the mouth, lower face, and the temple. So it's our purple area in that diagram. This is also where we have the efferent bit, the separate branch that's a motor branch going out to the jaw to help us to chew. So nociceptors of the trigeminal nerve innervate the meninges and the meningeal arteries. So this, in other words, is where transduction occurs. The interaction of these nociceptors of the trigeminal nerve and the meningeal vessels is known as the trigeminal-vascular system, or TVS. All three trigeminal branches, we've just talked about those three branches, enter the pons via the Gasserian ganglion. The Gasserian ganglion may also, you may see it in diagrams as the trigeminal ganglion or the semilunar ganglion. It's the equivalent of the dorsal root ganglion that we've talked about before, about nerves entering the spinal column, the spinal cord. This time, however, we're entering the pons. So transduction at the meninges and the vessels therefore transmits action potentials, if they're summated at the trigger zone, along the trigeminal afferent via the Gasserian ganglion and into the pons. So let's take that all the way through and up to the thalamus and talk about transmission. We have transduction at the meninges and the vessels. As we said, if enough EPSPs are created, they're summated at that trigger zone, just before the free nerve endings. And if an action potential is generated, it's transmitted along the primary afferent of the trigeminal nerve via the Gasserian ganglion and into the pons. Once in the pons, the primary afferent drops down, so it actually descends for a little while before it ascends again. It drops down into the medulla. In the medulla, at an area known as the trigeminal nucleus caudalis, the TNC, the primary synapses at multiple levels with the second order afferent. The second order afferent decussates, so switches over to the contralateral side and then ascends via the contralateral trigeminothalamic tract. Ultimately, it reaches the thalamus.
Okay, so as we said, the trigeminal nerve is primarily sensory. It has both unmyelinated C fibers and myelinated A delta fibers. All three branches enter the Gasserian ganglion, and this ganglion, like our dorsal root ganglion, contains the somata, the cell bodies of the trigeminal nerve. From the ganglion, the primary afferents enter the pons, and then they drop down, they descend on the same side to the ipsilateral, same side, trigeminal nucleus caudalis, TNC, in the medulla. And again, the TNC is an extension of the dorsal horn. In terms of the trigeminal nerve, it's our dorsal horn equivalent, but it's in the medulla. In the TNC, these primary afferents synapse at multiple levels with second-order afferents. The second-order afferents cross over, decussate, and then ascend via the contralateral, the opposite side, trigeminothalamic tract up to the precortical relay point, the thalamus. At the thalamus, the second-order afferents synapse with third-order or tertiary afferents, and of course they then ascend via the internal capsule to the somatosensory cortex. So that was our ascending, with a slight descent, pain pathway. So that's what gives us the pain. But of course, we've also, whenever we're talking about C fibers in particular, we have to consider the possibility of neurogenic inflammation. With that comes also sensitization of both the peripheral nociceptors and central synapses. In this case, what is also quite key is that it comes with vasodilation. So we've talked about our trigeminal afferent going off from those meningeal vessels via the pons to the TNC, and then the second order afferent ascending up to the thalamus and the somatosensory cortex. But C fibers, and in this case A delta fibers, secrete neuropeptides from specialist terminals when stimulated. So if you think about it, what are we talking about here? We're talking about our neurokinins, our neuropeptides. We're talking about substance P, calcitonin gene-related peptide, and neurokinin A. Although in this case, calcitonin gene-related peptide is the most important one in terms of outcome. So, calcitonin gene-related peptide, the important thing about it is it's a potent vasodilator. So it's secreted from those specialist axon terminals when the trigeminal nerve is stimulated. It comes out, it binds to smooth muscle cells, and it causes vasodilation. Substance P does help. It degranulates mast cells, and of course mast cells then release the vasodilator histamine. This does have a weaker effect than calcitonin gene-related peptide, but it still contributes to vasodilation. Not only do CGRP and substance P cause vasodilation, of course they also cause neurogenic inflammation. They upregulate. Histamine is an inflammatory mediator. Plus they stimulate others. Our mast cells will start secreting prostaglandins, all sorts of things. So they cause neurogenic inflammation. They also cause sensitization of the peripheral nociceptors first. So the little channels on the peripheral nociceptors will open more readily, but ultimately also sensitization of the synapses within that ascending pain pathway. So we've got vasodilation, inflammation, and sensitization. That brings us on to another way of causing vasodilation. This is the more of a neural way rather than the chemical way. This is the trigeminal autonomic reflex. And it's going to involve, as you might be able to tell from the diagram there, the facial nerve. So we have a functional connection between the trigeminal nerve and the facial nerve. The trigeminal being mainly afferent and the facial being mainly efferent. So the facial nerve has motor neurons that are responsible for facial expression. It does also have sensory neurons to the ear and tongue, but more importantly, in terms of vasodilation, it has a parasympathetic outflow. In other words, it has parasympathetic efferents. The parasympathetic efferents manage to stimulate salivation, nasal mucus production, and lacrimation. So they affect, we get a parasympathetic outflow in terms of all the glands of the face, which causes an increase in secretions. In terms of our vasodilation, acetylcholine, if you remember, in lots of other places, we don't have a parasympathetic outflow affecting vessels generally. What we're talking about is more of a sympathetic influence on vessels, vasoconstricting them. In this case, acetylcholine does cause vasodilation. In addition to this, the parasympathetic efferents also release other vasodilators. So in addition to acetylcholine, they will release vasoactive intestinal polypeptide, VIP, and nitric oxide. You may wonder what an intestinal polypeptide is doing up there, but it's probably something to do with where it was first discovered. So we have ACH, VIP, and nitric oxide NO, all causing vasodilation when the facial nerve is stimulated. So we have a parasympathetic outflow from the facial nerve, which increases vasodilation of the cerebral vessels. So how do we activate the facial nerve? What is this functional connection between the trigeminal and the facial nerve? Well, we've talked about our trigeminal. It transduces its action potentials at the meningeal vessels. The afferent then goes in via the pons, drops down to the TNC. It then synapses with its second order afferent, which then ascends on the contralateral side via the trigeminothalamic tract to the thalamus, and then ultimately via the tertiary afferent to the somatosensory cortex. So that's transmitting that pain series of action potentials. So that's transmission and transduction. But the second order afferent also branches to something called the superior salivatory nucleus. So in other words, we generate an action potential down here. It travels all the way up our trigeminal primary, drops to the TNC, is chemically transmitted at this synapse for the second order afferent, goes along the second order afferent, and yes, it will ultimately ascend. But in passing, it will also be transmitted along this branch into the superior salivatory nucleus in the pons. There's our superior salivatory nucleus. When this happens, of course, we're going to get an outflow, if you like, from that second order afferent to the superior salivatory nucleus. So, if that little branch ascending up there back to the pons synapses with a parasympathetic efferent of the facial nerve, we're then going to transmit it on and back out. So it's a classic reflex, isn't it? Because we've not gone anywhere near the cortex. We've got a reflex action where what's coming in via the afferent is connecting and synapsing and transmitting with an efferent. In this case, the parasympathetic efferent of the facial nerve. That then synapses itself. It synapses at this ganglion, the pterygopalatine ganglion that you don't really need to know about, but you might see it in diagrams, so I've shoved it in. What's important here is it's going to then get passed on to this postganglionic parasympathetic efferent down to these axon terminals that are very, very close to the meningeal vessels. Once they're there, that action potential is going to open voltage-gated calcium channels, and we're going to exocytose ACH, vasoactive intestinal polypeptide, VIP, and nitric oxide, NO. All are 3 vasodilators. So consequently, this neural pathway is going to cause vasodilation in the same way as our neuropeptides, our calcitonin gene-related peptide, did chemically. So let's talk about migraine. Only about a third of migraine sufferers get the classic aura. The aura can involve transient focal neurological symptoms, visual disturbances, in other words, like the scintillating scotoma you can see there. Sometimes you can get a unilateral paresthesia or dysphasia, various other sensory abnormalities. Migraine with aura, MA, and without aura, MO, are variants of the same disease, and individuals can have both. So they can, on one episode, they can have an aura, on the next episode, they might not have an aura. Or there have been cases where people have gone on to develop migraine with an aura or without an aura. So it can vary within the same individual as to whether you get an aura or not. Migraine with aura is more likely to be genetic, whereas without aura is more likely to have an environmental component. So we've got this same thing again. We've got a mix of environmental factors and genetic factors. But about half of migraineurs, in other words, people who suffer with migraine, do have a family history in a close first-degree relative. And there are thought to be multiple genetic mutations causing a predisposition to migraine. And we'll talk later about channelopathies as a trigger, as a pathogenesis. Environmental triggers are often related to light, sounds, smells. I know I used to get migraine and mine seemed to be triggered by hypoglycemia. So yeah, it wasn't chocolate. Chocolate was the cure. That's a good migraine to have. Various foods, though, have been implicated, including chocolate and cheese, and various theories about the chemicals within them that trigger the migraine. The weather has also been implicated. So there's lots and lots of different triggers. Okay, so the aura. The aura is caused by something called cortical spreading depression. It's agreed that this, at least, is the cause of the aura. And also, there are various theories and ideas are changing constantly about this. And one of the more recent ones is that it's been linked to raised extracellular potassium. So, in other words, that gradient in these people between the intracellular and the extracellular in terms of potassium concentration is different. What happens in CSD? You get initially only lasting for a few seconds, 2 to 5 millimeters per second is its travel rate, and that's only across half the cortex. You get a wave of depolarization. So although this is called depression, the first wave is about hyperexcitability. So you get a wave of depolarization, travels across the cortex, usually only half the cortex, at a rate of 2 to 5 millimeters per second, so very quick. This is then followed immediately by a few seconds of increased blood flow, increased regional cerebral blood flow, RCBF. And this is down to a brief period of vasodilation. So again, this is a very rapid, short duration wave. Depolarization followed by an increase in regional cerebral blood flow due to vasodilation. What happens next is the depression component. So for about, again, not a long period, for about 5 to 15 minutes, you get a reduction, a hyporeactivity, if you like, a reduction in synaptic activity within that area. What comes with that, though, is hypoxia. So you get 1 to 2 hours of hypoxia. So you get reduced regional cerebral blood flow. And in this case, it's due to vasoconstriction. And you also have a lack of availability because you can imagine, we've had this wave of depolarization, we've got all this switch round of ions. We need to repolarize, we need to get back to resting membrane potential. So the sodium potassium ATPase pumps are going to be going nuts. And of course, as we know, they need ATP to do that. So they will be using the available oxygen to make ATP to run the sodium potassium pumps to try and repolarize all those neurons. So there is an overuse, if you like, of available oxygen, but there's also a restriction in available oxygen because of vasoconstriction. And this period lasts for a couple of hours potentially. Now, the reason in all these diagrams it's gone the way it's gone is because CSD often starts in the occipital region, in the visual cortex, at the back of the cortex. It then usually stops at the central sulcus, so the big crease down the middle of the hemisphere, if you like. And it's usually confined to one hemisphere, so it's unilateral. Sometimes, though, you can get bilateral, and this is something called bacillar migraine, because it starts in the brainstem. If it starts in the brainstem, it tends to affect both sides. Now, CSD isn't particular to migraine, and on Moodle I've got an article for you that looks at CSD in other conditions. So it also occurs after CVA, when there's any trauma to the brain. So it's not just seen in migraine. So in addition to this hypoxia, we get this soup of chemicals. We get a complex and abnormal chemical profile in the brain because we have ions, we have solutes, all collecting, all being released, trying to be readjusted, rebalanced. So we get extracellular accumulations of ions, so might be protons, might be potassium. We also get solutes such as nitric oxide, adenosine and ATP. Of course, they're responding to vasoconstriction by being released to try and vasodilate to compensate. Glutamate causes the neurotransmitter in the CNS. When you get depolarization, there is going to be a lot of glutamate released. And potentially, if you release a lot of glutamate, trying to then mop that back up again could take a little bit of time. So we've got all these excitatory neurotransmitters, these vasodilators, these ions all floating around. Hypoxia and these solutes are what's thought to trigger the aura symptoms. So the sensory disturbances, such as scintillating scotoma or hearing problems, they're thought to trigger those and give you the symptoms of aura. They also stimulate the trigeminal nociceptors. So where they've been released on one hemisphere, they will stimulate, irritate those trigeminal nociceptors, will have transduction of those trigeminal nociceptors, which will then be transmitted by the trigeminal nociceptive pathway and will give headache. Because you've stimulated those trigeminal nociceptors, there will be neurogenic inflammation. Part of this neurogenic inflammation, of course, is sensitization. So the peripheral nociceptors and central synapses will become sensitized. Generally, that happens fairly quickly, surprisingly quickly, and this will increase the pain transmission. We're also likely to get meningeal vessel vasodilation. So calcitonin gene-related peptide, of course, causes vasodilation in its own right, and then substance P, SP, degranulates mast cells, and histamine, of course, is a vasodilator. There may also be that neural pathway leading to vasodilation due to the trigeminal autonomic reflex, that parasympathetic outflow to the meningeal vessels. So we get ACH, VIP, nitric oxide, all causing vasodilation of the meningeal vessels, plus our neuropeptides, our calcitonin gene-related peptide and substance P. Vasodilation and sensitization cause increased trigeminal stimulation. So, of course, the trigeminal nociceptors are sensitized. They're going to produce EPSPs far more regularly, far more quickly, which is going to increase the chance of action potentials. Those action potentials are then going to be more easily transmitted at synapses because of central sensitization. The vasodilation itself, plus all that chemical soup, are going to irritate the trigeminal nociceptors. Okay, so we've talked about aura, and you can see why that might cause migraine. But the problem is there's also the people who don't have an aura. So is cortical spreading depression a trigger for migraine, or is it just responsible for the aura? And as I say, what about the 60-odd% of migraineurs who don't get an aura? What's happening there? Is there such thing as a silent cortical spreading depression? So these people don't get the symptoms of aura, but they do have cortical spreading depression. And this, they managed to do a PET scan on someone that showed that someone who wasn't experiencing, wasn't perceiving any symptoms of an aura did have alterations in the regional cerebral blood flow. So it looked like, yes, they were having cortical spreading depression, but it was silent. They didn't have the symptoms of the aura. But that was, so far, only one study. And I mean, it used to be thought that migraine was a sort of vascular disorder, and that the vascular changes involving vasodilation of these meningeal vessels was the primary pathogenesis. But now theories have moved away from that, and now it's not thought to be primarily vascular. Yes, there are vascular changes happen, but that's not the actual pathogenesis of migraine. So there are lots and lots of theories on it. The other thing we said about the ions and solutes following CSD, that seems very logical. But again, it brings us back to this question of what if there's no signs of an aura? Are they having CSD? Okay, so let's look at some of those theories. So the first one looks at those ions and solutes and questions their validity. So ions and solutes, the ones collected after cortical spreading depression, this soup, as we said, are generally thought to stimulate the trigeminal nociceptors and cause the pain of headache. But other scientists have looked at this and they've said, well, no, actually, they'd be cleared away before they could activate the trigeminal nociceptors. And they looked at this and they demonstrated that these ions and solutes would be cleared away within 14 to 25 minutes. And they're saying that's not enough time to stimulate the trigeminal nerve. So this is where our PANX1 or Pannexin-1 megachannels come in. And these are activated when there's intense depolarization and overactivation of those NMDA channels. If you remember, in central nervous system synapses, you have AMPAs and you have NMDAs. AMPAs are our straightforward ion channels. The NMDAs, if you remember, need depolarization intracellularly from the AMPAs. They also need the binding of glutamate and glycine to open them. They're the ones with the little magnesium plug, if you like, that gets removed. And they only get stimulated in sort of fairly strong stimulation. In this case, when they're overactivated, they are saying that this activates these PANX1 channels in neurons. Now, PANX1 channels are basically gap junctions. They're exactly like our old gap junctions we've talked about before, but instead of the proteins being the protein connexin, the proteins are pannexin. So it's thought that these PANX1, when they're activated, in various ways, they increase the neurotransmitter release. And this increase in neurotransmitter release sustains cortical spreading depression. So it stimulates cortical spreading depression and maintains it. But it also stimulates the release of other things, including interleukin-1, our big bad boy, inducible nitric oxide synthase. In other words, we get nitric oxide and other pro-inflammatory mediators, including calcitonin gene-related peptide. So you can see we've got a couple of vasodilators there. We've got an increase or a sustained effect of CSD because of these PANX1 channels. And what they're saying is this sustains trigeminal stimulation beyond the removal of the ions and solutes. As I say, this is one theory. And I said to you before, when we were talking about the genetic component, I said there are channelopathies. Lovely word, that, channelopathies. Basically, what we're talking about is genetic abnormalities in channels. So if you think of how complex a channel is, and you can see a couple there, I've got a potassium channel there, fairly complex, TRPV1, so our vanilloid 1 channels, TRP channels, that's the one that responds to capsaicin, are highly complex structures. So if you change just one nucleotide in a gene coding for that channel, you can change the structure of that channel. So it may be dysfunctional. And this is the theory. There are dysfunctions of ion channels caused by genetic abnormalities. And many of these have been associated with migraine. In other words, they've noticed a correlation between people with these channelopathies and people who suffer from migraine. And about 112 voltage-gated cation channels so far have been seen to be dysfunctional in people with migraine, which sounds fairly convincing. So, of course, if they're dysfunctional or overactive, you're going to get changes in the ionic profile. And this change in the ionic profile could cause hyperexcitability. If you have a section of your brain that's hyperexcitable, it's going to be more prone to depolarization. And remember, CSD starts with that wave of depolarization. Everything else follows on afterwards. The other thing, of course, and part of the reason I've got a TRPV1 there, is the trigeminal afferents are stimulated by ion channels that are associated with our TRP receptors. These are our main superfamily, if you like, of receptors that create EPSPs. And remember, we learned about acid sensing ion channels, purinergic channels. So we have lots of other channels that will all create EPSPs. Those EPSPs get summated, we have an action potential that's transmitted up the pain pathway, we have pain. So the trigeminal afferents, their ion channels could also be dysfunctional. So there could be channelopathies that cause hyperstimulation of the trigeminal nerve. Which brings us to the neurolimbic theory. So what they found is that people who suffer from migraine, even between attacks, the interictal state, they have areas of neuronal excitability, specific areas. And it's thought that these can initiate spontaneous depolarization. So what they're saying is if these areas are hyperexcitable, then they can be like, if you like, the little firecracker amongst the bag of fireworks that sets off spontaneous depolarization, which then leads to cortical spreading depression. And part of this is thought to be central sensitization in the periaqueductal grey. Now you've heard of the PAG before. This is in the midbrain. And the rostral ventromedial medulla, RVM. And what this does, if you remember, we had the descending pain pathway. If you remember modulation, it was the phase, one of the phases of our pain pathways that we talked about in the module prep. If you can't remember it, you probably need to go back and have another look. So the descending pathway is supposed to be modulatory. It's supposed to block pain. It's anti-nociceptive. But at various points, we have this complex interaction of interneurons. And these interneurons act as on and off switches. And what they're saying is that if there's changes in that descending modulatory pathway either doesn't work as anti-nociceptive or it can even become pro-nociceptive. So it reinforces pain rather than blocking it. And this is where the limbic system comes in. So what they're saying is both the PAG and the RVM have neuronal links, so lots of little interneurons all linked together, to the hypothalamus and the limbic system. So this is where the link comes in with the common triggers that people report. The triggers processed or regulated by the hypothalamus and or the limbic system, so we're talking about emotions, all sorts of things, can trigger spontaneous depolarization due to these neuronal links with the PAG and the RVM. So that descending modulatory pathway has now become almost an independent pathway in its own right that stimulates pain. So just in case you can't remember, here's our periaqueductal grey. Let me highlight it for you. And there's the rostroventral medulla. So in the PAG and the RVM, interneurons can be activated that switch the anti-nociceptive descending blocking effect to a pro-nociceptive effect. Consequently, pain is stimulated or reinforced rather than the afferents being inhibited. And this is where we get chicken and egg, of course, because other researchers believe that, of course, changes in the PAG is an outcome of migraine rather than the generator. And this also makes sense because, as we know, this making that descending anti-nociceptive modulatory pathway pro-nociceptive is part of chronic pain. It changes. The more it's stimulated, the more it's likely to change and start becoming pro-nociceptive. So it could potentially be an outcome of repeated migraine attacks rather than the thing that existed in the first place to cause the migraine attacks. So in other words, cause and effect has not been established. And most of these theories, to be honest, they are about correlation as opposed to cause and effect. When we start to talk about research, I'll talk to you about correlation. It doesn't prove cause and effect. As I said, if you can't remember the descending modulatory pain pathway, or the ascending pathway, God forbid, was a bit strange, then please do go back to the module preparation videos or handouts and have another look. Okay, so another one that focuses on sensitization is this sensitization by calcitonin gene-related peptide. We've sort of alluded to it a couple of times, but we've said that apart from being a vasodilator, CGRP sensitizes peripheral nociceptors and central synapses. So those central synapses are going to be at the dorsal horn, or in this case, in the case of the trigeminal nerve, the TNC, and at the thalamus before the tertiary ascends to the cortex. So at peripheral nociceptors, it sensitizes channels, CGRP, increasing the prevalence of EPSPs in the free nerve endings. And if you remember, we have our trigger zone between the axon and the free nerve endings, and that's like our axon hillock. It's going to summate those EPSPs. If the more EPSPs we have, the more likely there will be an action potential transmitted up the primary afferent. At synapses, calcitonin gene-related peptide is exocytosed from the presynaptic membrane of the fibers, both C fibers and A delta fibers. It diffuses across the synaptic cleft in the same way as neurotransmitters do, and it binds to its own receptors, CGRP receptors, on the postsynaptic membrane. So exactly like, really, a neurotransmitter. It then activates, of course there's going to be a G protein involved there, adenylate cyclase, cyclic AMP. Cyclic AMP then activates protein kinase A, and this phosphorylation by protein kinase A phosphorylates the NMDAs and the AMPAs. And phosphorylation of channels, of course, often will sensitize them, will make them open more readily. So CGRP binds to receptors on the postsynaptic membrane, ultimately resulting in phosphorylation of those channels, the AMPAs and the NMDAs within central nervous system synapses. It's also thought to drift off and bind to astrocytes. Now remember, astrocytes sit alongside central nervous system synapses, and they regulate the amount of glutamate available. And it's thought that our calcitonin gene-related peptide binds to the astrocytes and increases the availability of glutamate. So, of course, more glutamate, more EPSPs created. So the postsynaptic channels are more likely to open when bound, and there is an increased availability of glutamate in the synaptic cleft. Both increase the strength and frequency of EPSPs in the postsynaptic soma. And in this case, those EPSPs are going to be summated by the axon hillock of the postsynaptic neuron. Hypoxia is the last theory we're going to talk about in any detail. We'll talk about serotonin in a minute, but the research on that is just so unfathomable. Hypoxia, there's a lot of correlations. Hypoxia, so below 90 millimeters Hg arterial, increases regional cerebral blood flow, and it does this primarily through the use of vasodilators, the usual regulatory vasodilators, nitric oxide, maybe calcitonin gene-related peptide, and adenosine. If you remember, adenosine is a vasodilator. There are also, the minute you start to have less oxygen available, cells will switch and will try and compensate. so there will be increased glycolysis. This increased glycolysis could perceivably disrupt the blood-brain barrier and also stimulate the production of free radicals like ROS, which of course is quite damaging. In hypoxia, lactate is produced rather than pyruvate during anaerobic glycolysis. Lactate is what stimulates the adenosine release from cells. Some studies, there are quite a few of them. Some have presented evidence that claims that hypoxia causes cortical spreading depression, or in other studies that it stimulates migraine without aura. So there's studies that say hypoxia causes migraine with aura or without aura. And part of their reasoning for this is that in migraineurs who go up mountains, so in other words into high altitudes, they are more likely to get migraines with visual disturbances, in other words with aura. But again, it is just a theory.
So serotonin, okay, research is conflicting. The role of serotonin, we don't really know, other than it does have a role. Maybe more research will come out in a few weeks. Anyway, that sort of sums up the whole approach really to migraine, doesn't it? Because the thing is, there doesn't appear to be any unified theory in terms of the pathogenesis. And this sort of begs the question, is migraine just a single syndrome? Or is it multiple syndromes that have similar presentations, but all have a different pathogenesis? Whatever the trigger is, the important thing is that the nociceptive component, what is being stimulated, is the trigeminal nerve, the afferent trigeminal neurons that are causing pain. And we do know that cortical spreading depression is the cause of the aura. As I say, whether it causes migraine or not is a big question.